In this lab, I perform an extraction of lactate dehydrogenase. First, I prepare an ice cold extraction buffer by adding 2 mercaptoethanol, PMSF, and EDTA to 10 mmol trees. Next, I mince about 50 grams of chicken breast, remove connected tissue and fat, and record the sad mass. Then I disrupted the tissue by homogenizing it, blending it with 75 milliliters cold extraction buffer using 4 30-second bursts. This process makes cells to release protein like LDH, and cooling between bursts prevents denaturation. Next, I transfer a homogenized mixture into 2 pre-chilled 50 ml centrifuge tubes and place them at 9,000 G-force for 20 minutes for centrifugation. This process separates components by density, where heavy cells debris forms a pellet while soluble proteins remain supernatant. After centrifugation, I filter mixture through cheesecloths to remove lipid from the solution, record the volume supernatant, and save 3 aliquots labeled crude homogeneity. For the second part, we perform ammonium sulfate precipitation, also known as salting out. I slowly added 0.39 grams of ammonium sulfate per milliliter of filtered supernatant over a 15-minute period. To protect the enzyme, the supernatant was placed in a beaker sitting in an ice bath on a stir plate. I add a stir bar and mix gently, being careful not to create a vortex since vigorous mixing or boiling can denature proteins. The ammonium sulfate competes with proteins for water molecules, reducing protein solubility and causing them to precipitate. Under these conditions, LDH should come out of solution. After 30 minutes of stirring, I centrifuge the sample for 20 minutes at 9,000 G-force, discard the supernatant, and cap the pellet with concentrated LDH and store it at 4 degrees Celsius on the next lab. This was the end for protein extraction.